Hi, my name is Jamie and I'm going to be reviewing some of the material from lectures 53 and 54 for the final exam. So to start, we're going to start with lecture 53, which is on DNA recombination. Just a general definition of recombination is moving sections of DNA from one position in the genome to another distinct location. And this is largely responsible for genetic variation. So first, I'm going to review how recombination occurs during meiosis. Just as a general review, meiosis is an important part of the process of sexual reproduction and is responsible for the genetic diversity in sexually reproducing populations. So it begins with a diploid cell which has 2 copies of each chromosome and ends with 4 haploid daughter cells. So before any divisions begin, synapsis occurs, which is when the homologous chromosomes, which at this point is made up of 2 pairs of sister chromatids because there was a round of replication. So the homologous chromosomes line up with each other, making a synaptonemal complex. And during prophase 1 of meiosis, crossing over occurs, which is the exchange of genetic material between homologous chromosomes. So the Rec-BCD complex, which is 3 proteins with helicase and endonuclease activity, unwinds the helix and creates the strand breaks. We're going to start here. So first, endonuclease nicks the single strands. We'll just say it nicks them right here. And it only nicks 1 strand from each pair. So next is the Strand Invasion, where the recombination joint is formed. So I have that depicted here. This is the Strand Invasion, where this strand comes up and joins here, and this one comes down. Just in case you can't see the colors that well. And this is what I was talking about when I say the recombination joint. So RUVA and RUVB proteins catalyze the branch formation. This is the branch. And the REC-A protein then catalyzes formation of a holiday structure, which I drew over here. So the holiday structure can be resolved, meaning split back up into 2 linear double strands with the help of RUVC protein. This is an endonuclease, and RUVC can cut the structure horizontally or vertically. So in this case, if the endonuclease were to cut the structure vertically, the 2 strands that were not originally nicked, so this one and this one, would be cleaved and ligated together to form heteroduplexes and recombinants. So this is the first picture. It's a little different from the lecture because I thought that was a little confusing. So you can see how if it was nicked here, we'll say it's just after B, and then here, this is where these 2 would be ligated together and these 2 would be ligated together to form this. This is a heteroduplex and also recombinant structure. So alternatively, if the endonucleases were to cut the structure horizontally, so like here and here, and then these 2 strands were ligated together, so this would be joined to this, and this would be joined here, then this would form. These are called patch recombinants because there are little patches of recombination. This is here and this is here, but other than that, it's pretty much the same, so they're not like true recombinants.